Okay, so next up we have Romesh Abeysuriya.
Very good.
From the University of Sydney, and he's going to tell us about how to track parameters using neural field theory.
Yeah, so it's actually quite interesting going after that talk. I'm going to be speaking today about my work using neural field theory to characterize brain states in terms of their physiology, and then to examine the evolution of the physiology and the brain states over time, and then some of the applications that this might have in terms of brain stability and seizures. So I'm going to start with a very high-level overview of what we want to achieve with this type of framework, and then the neural field model that we're using specifically in this study, and then go into how we use this model to represent normal and abnormal brain states and their activity, how we can fit this model to EEG data, and then track the evolution of brain states, and then potential applications for seizures. So at a very high level, we're broadly interested in representing brain states using physiology. And so I've got this sort of schematic diagram here with a set of axes, and I've labeled them here physiological quantity A and B. So at this stage, we're not so interested yet what they are, except that these are quantities that we want to change meaningfully or significantly with brain states. So for example, on one side here we have sleep, and on the other side wake. And then the other thing that we want from these quantities is we want to be able to relate them to dynamical activity that we could measure, for example, with EEG. And if we went to an individual's brain at a particular moment in time and then measured these quantities, we would see that at that moment, they would be at a single point on this plot. And then if we measured those quantities repeatedly over time, as their brain state was changing, we would see that they would trace out these trajectories on the plot over the sleep-wake cycle, or over other changes in brain state. And we expect to see different trajectories for different individuals, because we know that different brains are different, and there's a tremendous amount of variability even amongst healthy control subjects. But we'd hope to also see some common patterns emerge. So here, for example, we have large values of A being associated with sleep compared to wake. So where this picture sort of interacts with our traditional understanding of brain states, at least in the context of sleep, is that traditional sleep staging amounts to classifying regions of this parameter space, that might have similar dynamical activity, and then labeling them as one of a small number of these sleep stages. So anywhere in this blob here, you might see EEG activity that's typical of what you might measure in a subject who's awake with their eyes closed, and so on for the other brain states. So these states now correspond to the typical behavior, or the typical signals that we'd measure, but a superset of them would be a set of all healthy brain states, and I've labeled them stable here, but they could well just be labeled as healthy. So oscillations that are not associated with pathologies like seizures. And we might say that if we were sitting out over here, we might be a subject who has a particularly unusual EEG spectrum for whatever brain or whatever arousal stage they were in. And so this would reflect just normal individual variability. And then accompanying this classification would be a regime of pathological or unstable dynamics, and then we would associate this with seizure-like activity. So in terms of the transition from a healthy brain state into a seizure-like state, we would have a trajectory that starts out somewhere in this healthy regime, and at some point diverges. And then when it crosses a boundary separating our healthy activity from our seizure-like activity, that would be the point at which we would detect a seizure in the quantities that we're measuring, like EEG. So the overarching question then is, how can we make this schematic picture quantitatively? How can we link normal brain states, and in particular the sleep-wake cycle, with our understanding of seizures and seizure onset? So the approach that we've taken is to use a neural field model and then take some of the model's quantities for these quantities, these hypothetical quantities here, A and B. Many of you are already quite familiar with neural field theory, the broad idea being that instead of simulating individual neurons, we're going to simulate continuous fields that translate the properties of the individual neurons onto larger spatial and temporal scales. And this approach is particularly powerful when we're interested in looking at measurements of neural activity that intrinsically average over very large numbers of neurons, so in particular EEG. But there's another benefit of using neural field models compared to neural mass models, and that is that within a neural field framework we can incorporate spatial variations in the model parameters, which means that further down the line we can start to talk about how we can reproduce differences in EEG power at different positions on the scalp, and in particular for seizures, we can talk about local instabilities and localized seizures and then investigate seizure spreading within this same framework. So the idea is to use this one framework to bring together a whole lot of different phenomena. The particular model that we're looking at here is a quanticothalamic model with 4 neural populations. We have 2 in the cortex, a population of excitatory and inhibitory neurons, and then 2 populations of neurons in the thalamus. We have a population of thalamic relay nuclei that receive sensory input and then relay it into the cortex, and then we have a thalamic reticular nucleus that has inhibitory projections into the relay nuclei, and therefore it can play a regulatory role in this corticothalamic network. So, for example, when you go to sleep and we have less sensory input going into the cortex, that can be produced by an increase in activity in that reticular nucleus. And one of the most important things about this model in particular, and this modeling approach, is that all of the model parameters match physical, measurable quantities. So there's no arbitrary free parameters. Of course, our estimates for the parameter values, some are better than others depending on what experimental data is available. But whenever we have a set of model parameters that gives rise to an EEG power spectrum, for example, we're always able to relate that back to what that might mean for the brain. And that's a very powerful thing in terms of general modeling approaches. Another very useful feature about this model is that the EEG power spectrum can be generated from the model parameters with very little computational cost. And that's true for the healthy brain stage, which we can model by linearizing the system and then applying a perturbation, which gives us the power spectrum directly. And of course, that approach breaks down when we start to ask what dynamics do we predict during seizures. And so for those, we need to integrate the neural field equations directly. But certainly for the healthy brain states, we're able to use that linearization and do those computations very efficiently. And finally, it turns out that for this system, the dynamics can be well represented, not perfectly, but well represented by just 3 key feedback loops. So there's one loop within the cortex, another loop between the cortex and the thalamus, and that loop has 2 branches. There's one that goes through the relay nuclei and back, which is excitatory, and then another that goes through the reticular nucleus, then to the relay nuclei and to the cortex, which is inhibitory. And so this loop parameter y is effectively measuring the net contribution of the thalamus to the cortical activity. So there's this balance between excitation and inhibition. And then finally, we have a loop within the thalamus, just linking those 2 thalamic populations. So I'd like you then to keep in mind these 2 sets of, things. One is the 8 connection strengths, which are directional and connect the populations themselves. And then the other are those 3 feedback loops, X, Y, and Z, because many of the plots that I'm going to be showing you a little bit later on use those loop strengths as their axes. So the first question is, First is, can we use this model to reproduce normal EEG in all of the traditional arousal stages? And the answer is yes. So on the left I have typical examples of the EEG power spectrum in all of the traditional sleep stages, and then on the right, corresponding model fits. And we're able to do quite a good job of reproducing the key features in those spectra. So the frequency and relative size of the peaks that are present, the overall band powers. And so we then want to know, in the model, what are the parameter regimes that give us the activity that at a population level defines the traditional sleep stages? So to do this, we've gathered together quite a bit of experimental data. So in wake we have example EEG spectra for just over 2,000 subjects, and then for sleep we have 28 full night recordings that have been scored traditionally by expert scorers, and when we split them up, we end up with 20,000 examples of sleep EEG spectra. And then we go through all of these EEGs, and we can extract features from those spectra to characterize the states. So we're looking at things like the band powers, if there are any peaks present, and how big those peaks are, and the overall slope in the spectrum. And then we go to the model and test a whole lot of different parameters. And for each set of parameters, we compute the power spectrum and then compute the spectral features that we have in our model spectrum, and then compare them to the features that we saw in the experimental data to see if that spectrum was consistent with one or even several of the traditional arousal states. And when we do that, this is what we find. Here, the axes are corresponding to the strengths of those feedback loops I showed before, so that cortical, quanticothalamic, and intrathalamic feedback loops. And we can see now we have these colored blobs representing the parameters that give rise to EEG spectra that are consistent with those traditional sleep stages. So this is the quantitative analog of that schematic diagram at the very start of the talk, with those red zones corresponding to the traditional sleep stages. And then now we know what these axes are. They're the loop strengths in our model. So there's a couple of things to take away from this plot. The first one is that all of these states lie in a relatively small part of the model's parameter space, so they're all clustered together. And secondly, we can see that these states can be distinguished by these three axes. So we can see the different colored blobs, and they're separated in terms of these three parameters. But finally, these states overlap somewhat, which means that it's possible to transition from one state to another without ever leaving this zone of healthy, physiologically realistic activity. So if we had a brain state somewhere in here, and then the person fell asleep, they could travel through here, through stage 1 sleep, and then through to deep sleep without ever leaving this zone of healthy brain activity, which is exactly what we'd like to see. So this is then how we can use our model to understand our normal brain states. So what about the separation between the stable brain states and unstable brain states, between the healthy brain activity and the seizure-like activity? So if we make a simplification to the model, in terms of some of the synaptic, some of the dendritic effects, we can actually compute the stability for the model exactly in terms of these three loops. And what we've found is that this approximation actually generalizes quite well to the full model in terms of qualitatively and somewhat quantitatively giving us a good idea of what's going on in terms of the stability of the system. And what we find is that all of the steady states in the model lie underneath this surface here. So they're all clustered around there from the previous slide. And this, and then outside this surface, so like over here and in front of that space, we have pathological oscillations that are dominated by nonlinear activity and that we associate with seizures. So another interesting thing about this stability surface is that it's colored according to the frequency of the instability. So depending on where you exit the surface, you will see different dynamics in your seizure state, and therefore be able to produce different types of seizure-like activity. So the next question is, how can we compute the individualized trajectories for a particular subject that actually gives us information about a single person's physiology? So we can do this using a new fitting system that we've developed to map the model or to match the model power spectrum to the EEG, which gives us those individualized estimates, and then we can see the paths that those brain states trace out through the parameter space. So what we're looking at here on the right is the experimental EEG power spectrum in blue, and then in red our model fit. And then on the left-hand side, we have, again, our three loops, X, Y, and Z, the stability surface, and then the model parameters that gave rise to the spectrum shown on the right correspond to this red dot here. And now this EEG was taken over 5 minutes for a person going from sleep into wake. And if we set that going, we can see that as the EEG changes, the fit is evolving, the model parameters are changing, and it's also worth noting that the stability surface in the model depends on the model parameters, and so over time that surface is moving as well. And you can see as this person wakes up, their parameters are shifting from sleep into wake. We can see the alpha peak that's typical of wake states forming and then being fitted by the model. So we're able to recover pretty much all of the key aspects of that schematic diagram that I showed at the start. We know where our stability boundary is between the healthy states and the seizure states. We know where the traditional sleep stages are, and we can look at an EEG and estimate the trajectory that an individual's brain state has taken. So to consider the application to seizures then, let's examine, I want to examine synthetic trajectories in the model. So the way that I've come up with these synthetic trajectories is by looking at those zones in the model's parameter space and then picking points in terms of their connection strengths that produce EEG spectra that are typical of that stage. So each of these points is contained within that colored blob representing that sleep state. So these are typical examples of those stages. And then to produce the trajectories, what I've done is linearly interpolated the connection strengths between those two states. So we have our endpoint and then a bunch of intermediate states. And what we find firstly is that this linear interpolation produces curves in our space of X, Y, and Z. And then we can also look along the trajectory to see whether the brain state is stable and stays stable, and whether the firing rates and the power spectrum are consistent with what we see in physiology. And yes, for these transitions between the healthy brain states, that's certainly the case. So in terms of understanding seizures then, I want to focus on just one transition, which is from sleep state, as an example, from sleep stage 2 to wake with eyes closed. And I want to ask two questions. The first one is, can we reproduce this trajectory by integration of the nonlinear equations? Because this trajectory is produced using the linearized version of the model, which is fine for the healthy brain states, but not for the seizures, or not for understanding the dynamics during a seizure. And then secondly, I want to ask, what happens if we extend this trajectory? Because we can see it's sort of turning and heading towards the stability boundary. What if we went just a little bit further and crossed that boundary? So in this case, effectively waking up a little bit too much. So as we follow this particular trajectory, ending at our healthy wake state, what I've got here is the firing rate in the population of excitatory neurons. So this is the signal that we would associate with the EEG that you'd measure. And in red, we have our prediction of the linear stable state, the stable steady state in the model, and then in blue, the firing rate when we integrated the full nonlinear equations. And what we find is that provided that we change our connection strength slowly enough, the system continues to converge to that steady state. So it's a classic separation of timescales where we have our slow dynamics of the change in the steady state and our fast dynamics for the neuronal activity itself. And then if we continue this trajectory just a little bit further, what we see is that at some point our steady state vanishes as we cross that stability boundary in the model, and at that point, the firing rate starts to diverge, and then we start to get pathological oscillations that are nonlinear in origin and seizure-like. And it's interesting to note that this divergence into the seizure state is driven by the neuronal activity only after the stable steady state is lost. So once we're at about here, the firing rate it continues to increase and takes us into this seizure state. But up until that point, what's really important to understand the system's dynamics is what's happening to the steady state, not necessarily what's happening to the firing rate itself. So the changes that are occurring in the steady state could be modulated by other factors, such as chemical processes. So something quite different to the electrical activity that we're measuring. And if we visualize this trajectory in terms of the model's phase space, we can see that indeed, as we go from our sleep state to our wake state, the nonlinear integration gives us a path that more or less exactly follows that linearized trajectory. But then once we wake up a little bit too much and hit the stability boundary, then the dynamics diverge and we start to see large nonlinear oscillations. So how do we then relate this to seizure prediction and warning? So this is now looking down at the top. So we've got just an XY plane, and we can see our trajectory going from stage 2 up to eyes closed and then continuing a little bit further. And the thing is that the trajectories are continuous. So when we enter this unstable zone, we must have spent some time on the other side, quite close to it. So what we would do is we interpret the proximity to the stability boundary in the model as a risk factor for seizures and potentially an early warning sign, because loss of the steady state could be achieved by a transient excursion from stability. And so if we're sitting over here close to that stability boundary, it would take a smaller perturbation to cross it and then enter this unstable regime with seizure-like activity. So all of this area here corresponds to brain states that have an elevated risk of seizure onset, even if that stable steady state is still present and the dynamics of the EEG are still all right. So we can say that, as I mentioned before, exiting the stable zone from different parts of the model's parameter space can produce different seizure dynamics. So one possibility is that we could look at an individual's seizure and their waveforms and then infer perhaps which part of the model's parameter space they entered the seizure, and then from the dynamics before the seizure, it's possible that they always start having a seizure from the same part of the parameter space, which would mean that we could have an individualized seizure prediction zone. And then secondly, we could imagine using the velocity of that trajectory in order to predict seizures, because if the brain state is changing such that it's very rapidly approaching that stability boundary, maybe you're more likely to have a seizure sooner compared to if the brain state was changing slowly. And then finally, in this framework, the state changes are driven by the slow processes that were changing the steady states in the model. And then the seizure onset itself, in terms of the pathological electrical activity, is really a symptom of loss of that steady state. So this framework would certainly suggest that we could be targeting the slow mechanisms in terms of clinical interventions, and then tying into that the trajectories of the brain state prior to the seizure could perhaps provide some clues as to what those specific mechanisms might be. So to summarize then, we've linked seizure susceptibility to brain states in a unified framework that also models the sleep stages, and so we can relate the normal dynamics of the sleep-wake cycle to the pathological dynamics of seizure onset. And we're able to track the changes in brain states from experimental data. And we're able to actually do this in real time. And our work thus far has focused on the sleep-wake dynamics, and we're starting to extend this now to clinical populations. And we're excited possibly about being able to warn people when they're entering brain states that are more susceptible to seizures than others. And I'd like to conclude by saying that most of the ideas that I've talked about today are in this recent paper in the Journal of Neuroscience Methods. And I'd also like to acknowledge the support of the staff and students at the Brain Dynamics group at Sydney Uni, in particular Peter Robinson, Chris Rennie, and Svetlana Posnova. Thank you.
John.
Hi. I was just wondering if you could explain a bit more, first of all, how you linked seizure susceptibility to brain states in your conclusion slide, because I don't recall seeing any data at all relating to seizures in your talk. And then also, I was wondering, in terms of mapping this as a continuous path, lots of other groups, Wendling back in 2005, proposed this. So I just wondered, what's the actual novelty here? I guess it's that you're fitting these parameters to the power spectrum of the EEG system. Perhaps you could comment.
That's right. So to answer your questions in reverse order, the novel development in this work is the tracking routine that lets us infer the brain state in real time. And in particular, our advance is in incorporating temporal continuity, which stops that which stops our fitted trajectory from bouncing around too much in the parameter space, because it's a very high-dimensional parameter space, and it's possible that you could have multiple local minima or multiple acceptable fits. And so our development is being able to stabilize that by knowing that by imposing constraints on how rapidly the brain state can change. And of course, the ideas of mapping, of understanding where seizures are and where healthy states are in this sort of framework have been well established over the last decade. But you can't really use it unless you're capable of tracking, in terms of the clinical application, unless you're capable of tracking those brain states. And then in regards to the seizure susceptibility, I mean, it really comes back down to this slide, which is saying that if we're moving around through this parameter space, of course, if we're close to a stability boundary, then if you move a short distance in parameter space from here to there, you could be in a seizure, whereas if you were moving from here to there, then you wouldn't. And so that susceptibility is just related to the distance that you'd need to traverse in terms of changes to your brain state.
Stilly is next, and then Steve.
Well, as far as I understand, you're using this model basically to translate or to reduce the power spectrum parameters into few generated parameters within some neural field model. But the question is, what is indeed the added value of the dynamics of this model? Because you have tracing trajectories, but these trajectories, they are not autonomous. In a sense, there is no dynamics along this trajectory, and it's only dynamics on the specific point in the parameter space that generates the spectrum. And the second part of my question is, do you really think that the brain states are uniquely defined by the power spectrum of the EEG? I would expect there is much more than just power, like correlations between different sites or phase coherences or
Yes, those are both very important points. For your first one, you're absolutely right that we don't have a generative model for the changes that are taking place in those parameters, and so when we have that trajectory in terms of changes to the connection strengths between the populations, a model of how those should be changing. We've done some work of coupling a model, in terms of sleep, of coupling our model of the ascending arousal system to drive those changes in the connection strengths, but there is a lot more work to be done in terms of associating those connection strengths with particular mechanisms such that we could identify or predict in a sort of Kalman filter-esque way of what's going to happen next in the system, beyond what we're able to do at present, which is to look at the collection of subjects that we have and then associate and then infer the types of changes that we see in brain state based on what we have in the experimental data. And then for your second question, yes, we assume a lot by characterizing the brain state purely in terms of the power spectrum. But I think one of the things that we're most interested in doing is seeing how far we're able to get using just the power spectrum alone. And certainly in our work with sleep staging, one of the biggest challenges that we have, leaving aside just EEG alone, is that the power spectrum between wake with your eyes open and then REM sleep and sleep stage 1 is very, very similar. And so REM, for example, when you're dreaming, is primarily differentiated from light sleep by muscle tone, which of course is not measured directly in the EEG. So there is certainly an aspect of seeing how far we can get using the simplified approach, and then being able to ask what are the things that are essential to add on top of this to capture more brain states.
So that actually leads to my question. REM is very hard if you're only looking at cortical power spectrum to differentiate from being awake. They're not the same. And we know that there are some other nuclei that could be hooked into your model that are very clearly differential. But the only piece that I think you perfectly ought to capture here is the thalamic-cortical interaction spinning. But you made a decision to leave out the brainstem nuclei and the hypothalamic nuclei. You were involved in driving you into the state. But it didn't seem to matter. You seemed to get very good fits. You must have some thoughts about that. Is it okay to leave all that stuff out?
That's a good question. I mean, I think it comes down to how far you want to take a particular model that you have, having made simplifications to the system. And of course, we have done work in extending this particular model by adding populations of neurons in the basal ganglia, by incorporating bursting effects in some of the neural populations. I think it's interesting when taking this approach, really just to see how far we can get using this, using the simplified model, and then observing where that breaks down, giving us perhaps some insight into what are the most important remaining parts of the system. Certainly with REM, one of the things that we've found is actually quite interesting is that some of the higher frequencies in the EEG are affected by muscle artifact. So there's kind of an EMG component to the EEG power spectrum, and we see some interesting variations in that over switching between wake and REM compared to the other sleep stages. And where we have some, and our results are looking actually quite interesting at being able to use that component of the spectrum to get part of the way to distinguishing REM from the other sleep stages. And in regard to sleep spindles, and it's quite interesting, but sleep spindles actually come out quite nicely in our model because they seem to be primarily an intrathalamic effect, and that interaction between the thalamic reticular nucleus and the relay nucleus seems to be what's most important. And so I think we can do quite a good job of that.
I've got one final question. So you said you presented a beautiful theory, and you said you've tested it with some data. Does it work?
I guess it depends on what you mean by does it work.
Does it predict seizures?
No, and not yet. And I guess part of the reason is because we've developed this not trying to answer the question, can we predict seizures, but rather the question, can we characterize brain states in terms of their physiology based on existing measurements that we already have. And in regards to that, we set out to make a quantitative version of that schematic diagram at the start, where we could understand the changes that take place over the sleep-wake cycle, not in terms of changing between sleep stage 1 and sleep stage 2, but in terms of saying, my corticothalamic feedback strength has decreased and my intracortical feedback has increased. Mm-hmm. And in that respect, I think we've been quite successful.
Okay, thanks.
So right now